Okay, so here we have a DNA sequence. As you can see, our two parent strands are anti-parallel, meaning they go from 5 to 3 and then 5 to 3 on the opposite side. These two strands, between them the base pairs are bonded by hydrogen bonds, depicted by these dashed lines. These hydrogen bonds are then broken by the enzyme helicase that unzips the double helix. As the helicase unzips the double helix, the enzyme gyrase comes in and keeps it from supercoiling. Once the double helix is open, primase comes in and lays the RNA primer that starts the replication process. And it lays it in a 5 to 3 manner. Once the RNA primer is set, DNA polymerase 3 comes through and lays the DNA nucleotides and with their bonding pairs. And because this is the leading strand, the laying of these DNA nucleotides is continuous as the whole double helix unzips. DNA polymerase 1 then comes in and replaces the RNA primer with DNA base pairs that are again anti-parallel. Once the DNA is down, ligase comes in and forms a phosphodiester bond between the old DNA and the new DNA. Now onto the lagging strand, primase then comes in again and lays RNA primer in a 5 to 3 manner. Then DNA polymerase 3 comes through and lays the corresponding DNA nucleotides. This whole segment is called an Okazaki fragment that forms on the lagging strand, and they are fragments because they continue to form as the double helix unzips. Similar to the leading strand, DNA polymerase 1 comes through and replaces the RNA nucleotides with DNA nucleotides. Once they are placed, ligase comes in and forms a phosphodiester bond between the old DNA and the new DNA. Okay, the helicase continues unzipping the double helix. And gyrase comes in and keeps supercoiling from occurring. On the leading strand, DNA polymerase 3 continues placing DNA nucleotides with the corresponding base. Once the new DNA nucleotides are placed down, ligase comes and creates a new phosphodiester bond connecting the old DNA with the new DNA. To continue with the lagging strand, another Okazaki fragment is needed, so the primase comes in and places RNA nucleotides in the 5 to 3 order. And then DNA polymerase 3 places DNA nucleotides. Ligase then again comes and forms a phosphodiester bond between the old DNA and the new DNA. DNA polymerase 1 then again removes the RNA nucleotides in the 5 to 3 direction and replaces them with corresponding DNA. And ligase comes and forms a phosphodiester bond between the new DNA and the old DNA. And this is DNA replication.